Section 2, we will discuss the intestinal organoid technology. Intestinal organoids, broadly speaking, are a 3D in vitro model system that recapitulates many of the intestinal features of the in vivo intestine. It consists of a polarized epithelial layer, meaning that there is unique polarity, an apical and basolateral surface, and an enclosed functional lumen. Functional in this regard means that there's a lot of active transport into and outside the lumen. A lot of cellular signaling is maintained, as well as there is presence of differentiated and different cell types. Intestinal organoids offer convenient research tools, and when we talk about intestinal organoids specifically, we gain a lot of flexibility and utility in these culture systems to be able to answer a lot of research questions. As mentioned before, organoids recapitulate cellular complement of the in vivo intestine. It means that in general terms, you have intestinal stem cells and you have differentiated cell types like goblet cells secreting mucus. You have Paneth cells, you have active enterocytes that can absorb things into lumen, and there are a lot of inter and intracellular signaling pathways in these organoids. You are able to look at the function of the intestinal epithelium in a tissue that is outside of an in vivo context and entirely an in vitro culture system. Intestinal organoids are genetically identical to the source tissue or biopsy and are great at retaining the histopathology of the starting samples or biopsy. Organoid cultures also retain the genetic fidelity to their starting source. Intestinal organoids are able to be expanded and maintained long term, which allows you to perform experiments over time. Lastly, you can cryopreserve and biobank intestinal organoid cultures for a later use. Biobanking allows researchers to leverage genetic diversity of the population, leading to a more realistic preclinical data. In 2009, there was a pioneering publication by Sato and Claver's group. They published the first method for culturing mouse intestinal epithelial organoids. This is the first key publication of the intestinal organoid technology that we know of today. This publication sprang up from the same group that were studying the niche factors that make up the intestinal stem cell niche in the actual in vivo tissue, and they were able to mimic this in vitro and create in vitro culture conditions to generate intestinal organoids. The general process to generate adult stem cell derived intestinal organoids is to isolate the crypts from the adult tissue, and crypts are the region of the intestinal epithelium that contains the stem cells, and then embed these crypts into an extracellular matrix and expose them to the media conditions that contain stem cell niche factors. These are essentially the growth factors that are present in the crypt domain that will help maintain the stem cell characteristics. In 2011, the same technology was published for human intestinal organoid cultures. The media was slightly different due to differences between human and mouse biology, but overall, there were very similar methodologies and principles between the mouse and human workflows. All of this is under a patent from Hub, whom STEMCELL have partnered with for exclusive global licenses for this technology. In section 3, we will discuss intestinal model and culture systems. There are several different model and culture systems used to model the intestinal system for a variety of research applications. The first model system highlighted here is the use of immortalized intestinal cell lines, for example, KECO2. The second commonly used model system is in vivo modeling using mice models. The third technique or model system commonly used is the in vitro culture of primary intestinal cells as organoids or organoid-derived monolayers. The last methodology involves the examination of surgical and cadaveric samples or tissue biopsies, which could involve histological assessment of cadaveric and surgical samples using H&E staining. The use of cadaveric intestine processed 12 hours post procurement represents an excellent alternative source of crypts, which can be used for in vitro culture of human intestinal organoids. This may offer a viable alternative source for labs or researchers with lack of access to fresh human biopsies. The following slide will focus on the highlighted or underlined methodology, which are in vitro culture systems, and provide a high level comparison between different approaches or methodologies. Immortalized cell line cultures, for example, CACO2, are considered to be a gold standard in the intestinal field. It is a highly published model system and is grown as epithelial monolayers. This allows ease of access to the apical surface, which can be advantageous for applications like infections. The limitation of this culture system is that it is a homogeneous cell population. Therefore, these monolayers cannot recapitulate the diversity of cell types in the normal intestinal epithelium. The cells are genetically abnormal as they are derived from tumor tissue and can proliferate and grow and expand indefinitely. However, they're not similar to the healthy intestinal systems as they usually harbor mutations. Organoid cultures are more physiologically relevant as the cellular composition is much more similar to that of an intact healthy intestine. Because you're growing these cells from a stem cell, they have the ability to generate different cell types of intestinal epithelium. Hence, you end up with cultures with these physiologically relevant intestinal cell types. Organoids can recapitulate the healthy and diseased phenotypes, but the key point is that the organoids have a genetic fidelity to the sample they are derived from. Therefore, modeling genetic diversity is quite straightforward with intestinal organoids, which is usually not the case when working with immortalized intestinal cell lines. The limitation of the organoid technology is the dependency on extracellular matrix such as matrigel. 2D organoid-derived monolayer cultures refer to monolayer cultures which are generated from organoids. This system combines the physiological relevance of organoids and 2D systems. This culture system can address the limitation of organoids, as in it allows an easy access to the apical side of the epithelium, which is important for certain research applications such as infections. This system also gives you an option of further differentiation using air-liquid interface. This is important for applications that require differentiated cell types like goblet cells, enterocytes, or enteroendocrine cells. One limitation is the loss of 3D structure or physiology. Also, it is generally considered to be an endpoint culture system, which means that monolayers cannot be maintained or passaged long term. This slide provides a high-level overview of different approaches for using 3D organoids for infection modeling. The first approach involves dissociation of organoids into single cells or small fragments, combining these dissociated cells with microbes and reforming organoids with infected cells. Technically speaking, this is the easiest approach. However, it can be difficult to fully model infection in real time, and it can be limited in how host pathogen interactions can be studied. The second approach involves microinjection, where bacteria or infectious agents are injected directly into the lumen of an organoid using a microinjector. Although technically possible, this is quite labor intensive and not amenable to high throughput applications. Lastly, organoids can be broken and seeded as monolayers. This system allows an easier access to the apical side and good differentiation of intestinal cells. Compared to microinjection, this method is more amenable to high throughput applications. Intestinal monolayers or air-liquid interface monolayers offer a versatile tool for viral infection modeling. Intestinal monolayers can be easily inoculated with virus or viral particles from the apical side, and different readouts can be obtained from apical side, basolateral side, and the tissue itself. Readouts from apical side would include morphological evaluation, tear measurements, ELISA readouts looking at cytokine secretion, and barrier function and permeability assays. Researchers could also evaluate the cytokine release from basolateral side and consider adding cells to the basal chamber for looking at immune cell co-culture studies. Readouts involving the tissue could include viral count, viral spread, for example, understanding how virus can move to neighboring cells or adjacent cells, immunohistochemistry or histological applications, ion channel activity assays, which could involve understanding the flow of ions in response to an infection, signaling pathway studies, and lastly, understanding innate immune responses triggered through innate receptors like toll-like receptors. Here is a brief summary of this lecture. The intestinal epithelium is primarily composed of 6 cell types arranged in a crypt villa structure. Intestinal organoids provide a 3D in vitro intestinal model systems that incorporate many of the physiologically relevant features of an in vivo intestinal tissue. These features include a polarized epithelial layer surrounding a functional lumen. Primary intestinal organoids can be generated by isolating intestinal crypts that contain adult stem cells. In 2009 and 2011, Sato et al. Published key papers describing how to generate intestinal organoids from intestinal crypts isolated from mouse and human tissues, respectively. Several alternative model systems are commonly used to model the intestine for a variety of research applications. The most prevalent include in vivo studies using mouse models and intestinal cell lines such as CACO2. Lastly, organoid-derived intestinal monolayer cultures can be a valuable tool for applications requiring access to apical surface. This concludes Lecture 1. You can now proceed to the Test Your Knowledge section to see how much you learned from this lecture before proceeding to Lecture 2.